Thank you for introducing me and for having me here presenting my work. So today I'm going to present a statistical method that we have developed called BASE. Can people in the back hear me clearly? Okay, I'll do my best. So the name of our method called BASE, and it's developed to perform a multi-scale and multi-sample analysis in spatial transcriptomics data. The two tasks our method is dealing with are the cell type clustering and spatial domain detection in the context of spatial transcriptomics. So here is a little bit background for spatial transcriptomic technologies. Spatial transcriptomic technology is a collection of many different methods that have been developed in recent years to collect two important information in a biological tissue. The first important information is gene expression information, or how actively a gene is going through the transcription and producing protein in the cell. And the second important information is the spatial location information, so that we know where the gene expression is happening in the cell. And actually, many of these technologies have been commercialized. For example, the Merfish has been commercialized by Visgene, Cosmics by Nanostream, Xenium by Tanx Genomics. And these technologies can roughly be categorized into two groups according to their technique. So some techniques are based on the sequencing technology, including the Visium, SliceSeq, and high definition spatial transcriptomic and SIG-scope. And these technologies, so different groups of technology has different features. And for technologies in this group, they usually have very high throughput, which means they can measure almost all the genes in cell. But usually the spatial resolution for these technologies are relatively low, which means for each spatial location, it usually contains multiple cells. It's not like cellular resolution or even subcellular resolution. And the other group of technologies are based on the single molecule fish technique. And these technologies include the Merfish, Xenium, and Cosmics. And an important feature of these technologies is that they're using microscope. So they can basically see the molecules under the microscope. So the resolution is under subcellular level. However, disadvantage of this group methods is that they can only measure like a few hundred genes because they are targeting those genes specifically. So to give you a sense like how these data are collected, how they are generated by the experiments, here I will give you two examples, one in each category, to show you like how this data is generated. So this is a very popular data called spatial transcriptomic data called Tanx Genomics. This is commercialized by Visium. So at the beginning, you have a so-called Visium spatial gene expression slide. And within this slide, you have 4 different capturing areas. And within each capturing area, you can basically take a slice of your tissue sample and place the tissue sample onto the capturing area to measure the spatial transcriptomics data. So within each capturing area, we have a lot of these spots. And within each spot is coated with a lot of primers. And this primer is basically a sequence of nucleotides that have different function units. For example, this poly T tail is designed to pair with a poly A tail in the mRNA. So it allows the primer to capture mRNAs, and also has a UMI component. The UMI stands for unique molecular identifier, and it's designed to debias the PCR procedure. And another important component is the spatial barcode. So after sequencing these primers, you can essentially know where this primer comes from on this capturing area. So this allows us to measure the spatial location information in this data set. And another group of methods are based on the fish technology. Here is an example. This technology is called Merfish, commercialized by Visgene. And so the idea of single molecule fish is basically you have a if I have a probe, it's a basic sequence of nucleotides, and it's designed to target specific transcripts. And in this probe, it's tagged with some fluorescent molecules. So after some trigger, you can actually see the color under the microscope. But the traditional SM fish technology, they can only target a few genes. So this MERFISH technology extends this traditional fish technology and do a series rounds of extinguishing hybridization of the probes onto the molecules. So at each round, some of the genes will be, you can see some of the genes, and it's coded to be 1. And for those genes that cannot be seen, it's coded to be 0. And after a lot of rounds, you have a basic barcode for each specific gene. So these barcodes are pre-specified before experiment. So you can basically use the barcode to identify which molecule, which gene this molecule belongs to, and then directly count the number of the molecules as the gene expression data. And for the location information, you can basically just see it under the microscope. So no matter which technology is used to generate the spatial transcriptomics data, it basically gives us 2 matrices. One is the gene expression matrix. It's a P by N gene matrix for N cells and P genes. And another important matrix is the N by 2 location matrix for N cells and X and Y spatial coordinates on tissue. And for some specific technologies such as the star map, it can measure the gene expression in 3D, so it also has a 3rd dimension. And with these 2 important data, we can perform many interesting analysis. And 2 specific questions that are of interest to many biologists are cell type inference and spatial domain detection. And we define a cell type to be a cell that has a unique gene expression profile. And we define spatial domains to be a region on the tissue that has a unique cell type composition. Here, I will use this synthetic cortex data to give you an example, like what our cell types look like and what our spatial domain look like. So in this syntax cortex, it has the different cell types. So we have excitatory neurons, inhibitory neurons, and different types of non-neuronal glial cells, denoted by different shapes and colors. And besides these different cell types, we also have 4 different spatial domains. We in this context, because this is cortex, so they're usually called layers. And we have layer 1 all the way to layer 4. And we can find that in each layer, we have different composition of cell types. Many methods have been developed to solve these 2 important questions, but unfortunately, they all have certain limitations. And the first limitation is that they focus on either one of the 2 tasks. They either do a cell type clustering or they do a spatial domain detection. However, just like I mentioned, these 2 tasks are interconnected with each other. Like if we know the spatial domain a cell belongs to because the spatial domain has a specific cell type composition, we can essentially use this composition as information to help us improve the inference of the cell type. And on the other hand, for the spatial domain detection, essentially the cell types are defining the spatial domains. And the other limitation is that most of these existing methods, they only analyze 1 tissue section at a time. However, for a typical spatial transcriptomic study, we usually take a series of cuts in the tissue. For example, here is a mouse brain, and we usually take a consecutive cut of the brain tissue and measure each of the slides in the tissue. And so in the end, we would have multiple tissue sections. And in certain cases, this is different tissue sections are measured in different subjects, like in different mouse, but in the same tissue type. So no matter it's measured in the same subject or different subjects of the same type, they are supposed to have they're supposed to have similar cell type composition and spatial domains. So we can essentially leverage this information across these multiple tissue sections to help us improve these 2 analyses. And the third aspect is more on a technical aspect. So most of the spatial domain detection methods, they specify a key spatial parameter, and they use this parameter to encourage smoothness across the spatial domains, because we assume the spatial domains are smooth. However, all these methods, they pre-specify this value, or they just rely on the user to specify the value. But actually, the final results of the spatial domain detection can be very sensitive to the parameter we specified. So for example, when the parameter is 0, basically there's no smoothness. You can see the segmented spatial domains are very messy. And if it's too large, you tend to over-smooth certain domains. Only when it's within an optimal range, we can have very good segmentation of the spatial domains. So given these limitations, we have developed a method called BAS, which stands for Bayesian analysis for spatial segmentation, that can perform multi-scale modeling. So we infer cell types at a single cell level and detect spatial domains at the regional level within a same hierarchical model framework. Another feature is that we can do multi-sample modeling. So that means we are jointly modeling multiple tissue sectors together. And finally, we can infer the key spatial parameter based on data at hand. So next, I will introduce our method. So our method takes 2 input data. Our method assumes that there are like capital L different tissue sections collected from a transcriptomic study. And we use lowercase l to denote one specific tissue section. And for each tissue section, we have a gene expression matrix, which is P by NL matrix for P genes and NL cells, and the spatial location matrix. And in addition to these 2 input matrix, we also assume that for each cell, for cells, they have 2 hidden states. So each cell has a cell type label CIL, and also have a spatial domain label ZIL. And the goal of our method is really to use these 2 input data to infer these 2 hidden states of cells. And we do so based on a hierarchical model. So we link the 4 components, the gene expression, cell type, and spatial domain and location information within a Bayesian hierarchical framework. And on the outermost layer, we have gene expression. And depending on the expression of that gene, we can infer the cell type. In expression profile of the cell, we can infer its cell type. And then once we know the distribution of the cell types, we can infer the spatial domains. And finally, we have the location information here to encourage smoothness and incorporate neighborhood similarity across the spatial locations. And next time we'll introduce like what type of models we use to model each of the relationship within this hierarchical model. So we use a multivariate normal distribution to model the gene expression features. So depending on the specific cell type the cell belongs to, the gene expression features follow a multivariate normal distribution with mean parameter mu c and variance covariance parameter sigma. And we notice that we know that this mean parameter is cell type specific, but this variance component, this variance parameter is shared across different cell types. And the reason is that we usually found that for some cell types, the number of cells is relatively small. So it's very hard to have an accurate estimation of this variance component, this variance parameter. So we decide to impose this constraint that let them be shared across different cell types to have a better estimation. And given the specific spatial domain the cell belongs to, we assume that the cell type of the cell follows a categorical distribution with a parameter vector pi r. Here pi r is the vector of probabilities of different cell types in that specific spatial domain R. And finally, we used a homogeneous plot model to incorporate neighborhood similarity and encourage smoothness across spatial domains. And these 3 models essentially link the 4 components, the gene expression, cell type, spatial domain, and location information together. This way, the inference of the cell type, the spatial domain, can be like connected together and end up helping us to improve the effectiveness of both of these analysis. So here is a little bit introduction to the POTS model. So POTS model is commonly used in Bayesian analysis as a prior to incorporate the relationship among variables. And it has two parameters. One is a neighborhood graph. This graph is basically tells you like what cells are the neighbor of each cell. And we construct this neighborhood graph using the spatial location matrix by searching for each cell the k nearest neighbors. And another important parameter, like I described before, is a spatial interaction parameter that controls the smoothness across neighboring locations. So the model and the algorithm based on the Bayesian framework. So we basically follow the standard Bayesian inference framework. We use the keep sampling and the Chauvet-Hastings algorithm to sample all the parameters from their for conditional distributions. But in this process, because we also want to sample beta and to finally infer beta, but this C of beta is a normalizing constant in this POTS model. It makes it really hard to evaluate this probability because it requires us to evaluate all the configuration of the spatial domains in order to get the normalizing constant. So we end up using an algorithm called Swenson algorithm, and it allows us to like sample beta more efficiently, and it also allows for a faster mixing rate, which means the samples are less correlated with each other. So in the end, we would have three output. One is the cell type cluster labels, and the second one is spatial domain labels, and finally we have the cell type compositions within each spatial domain. And this is basically our method. And next, I'll move on to the results section. Before I do that, are there any questions?
So, how do you get the ground truth label?
Can you speak a little bit?
How do you get the ground truth label?
We don't. It's a clustering, so it's unsupervised. It doesn't need ground truth labels.
So you still need to evaluate the performance of
we evaluated them in simulations, so we know the truth.
So there's no real.
We do have real data. We do have some real data, and you can look at anatomy of those tissues to like whether that makes sense or not, to see if that makes sense.
So for real data, can you do a ground truth label?
For real data?
Can you do ground truth label for real data?
We don't have the truth in real data analysis.
Okay.
Yes.
So with the like smoothness parameter, is that there because there's an assumption that there's like an error in determining like cell locations, or is that there just to like reorganize like the cell types into like specific places?
Because the interaction parameter is here because we want the neighboring cells, if they are located near each other, we want them to belong to the same spatial domain. That's it just basically allows us to incorporate this information into the model.
Okay.
Yeah.
Your quality expression data, why did y'all choose the multivariate normal distribution as opposed to something like SON or another distribution?
Like P distribution or other multivariate distribution? Are you thinking of T distribution or other distribution?
The question is, why did y'all choose the multivariate normal distribution to model the expression data as opposed to another?
Oh, we normalize the data, so it's more normally behaved. It's not count data. That's your question. So next I'll move on to the results section and talk about some of our simulation and real data application results. So So here is a brief introduction to our simulation design. So we used the real spatial locations of 1,127 cells from the mouse cortex data. And we assigned these cells to 4 different spatial domains. Just like the we'll show at the bottom, we have assigned cells into the layer 1, layer 2/3, layer 5, and layer 6. And we also, in addition to these 4 spatial domains, we also assume there are 4 cell types with different spatial distribution patterns. And for each spatial domain, we assume there are 3 cell types. And if we compare 2 domains, we have one unique, each spatial domain has 1 unique cell type and have 2 cell types that are in common. And we assume these cell types have different spatial distribution patterns. We designed a total of 4 scenarios. In the scenario 1, where the cell type has the strongest spatial distribution pattern, that means each spatial domain is basically dominated by 1 cell type. And in scenarios 2 and 3, the proportions of the dominant cell types are either 80% or 50%, and the proportions of the other non-dominant cell types are 10% and 25%. And in scenario 4, we assume it's a cell type has the weakest spatial distribution pattern, and each cell type is located in 3 different spatial domains. So this is the simulation results for spatial domain detection, and we compared our method base to 3 other commonly used spatial domain detection methods: the HMIF, AS-based, and SPAGCN. Because the HMIF requires the user to specify the spatial parameter. So basically, what we did is we specified a list of different data values and fit the model, and reported the results that give us to the worst, median, and best results for better comparison. And we evaluated the accuracy using the adjusted random index. So this index ranges from 0 to 1, and a higher value indicates a better accuracy. And we can find here, our method has overall the best performance, and it's followed by HMRF, SPAGCN, and the AS-based. And we can also notice that in scenarios 1 and 2, the performance of our method is very similar to the other methods. And this is because in these scenarios, the spatial distribution pattern of cell types are very strong. So for each spatial domain, it's merely composed of just 1 cell type. So for those cell types that directly model the spatial domain and don't consider the cell types, they can also have very good performance. However, when it comes to scenario 3 and scenario 4, where each spatial domain is composed of more different cell types, the performance of the other methods become worse. And for cell type clustering, we compared our method with 3 other methods: the SC3, SIRET, and FICT. And our method has overall the best performance, and it's followed by SC3, SIRET, and FICT. And one observation we can have here is that the performance of gain from our method compared to the other 3 methods is the largest in scenario 1. And this is because in scenario 1, the cell type has the strongest spatial distribution pattern, so it can benefit more from the spatial domain information. That's why it has have a larger performance gain. However, when the spatial distribution pattern of cell types become weaker and weaker, the performance gain also gets weaker. And besides the single sample analysis, we also did a multi-sample analysis by jointly analyzing multiple tissue sections together. So as you'd expected, if we increase the number of tissue sections, the performance of the spatial domain detection increases, and the performance of our method and the SIRET increases for cell type clustering as expected. However, for SC3, the performance increases first and then it decreases. Basically, it's because SC3 cannot handle very large data set, and when the number of cells exceeds a certain value, the performance just decreases. And a similar observation we can make here is in scenario when the performance gain is much larger compared to the other two. And this is because cell type has the strongest spatial distribution pattern. And the performance gain decreases as the spatial distribution pattern of cell types decreases. So when many people want to use our packages, come to us, commonly asked question is that how to specify the number of spatial domains and how to specify the number of cell types. And I think that's a common question for many like clustering methods. So we did a sensitivity analysis and evaluated like if we specify a round number of spatial domains, how this would change the performance of our method. So in our simulation, the true number of spatial domains is 4. And here along the axis, it's the specified number of spatial domains. And on the Y, it's showing the proportion of cells in each of the segmented domains. So as expected, if we under-specify the number of spatial domains, we end up only just having 2 clusters. And of course, many of the domains will be segmented into the same domain. And if we over-specify the number of spatial domains, one interesting finding we have here is that we still have largely 4 domains, and the remaining domains, what we call redundant domains, they only have very small number of cells in these domains. But for the other methods, like especially for Bayes space, it tends to split one spatial domain into multiple domains. So you can see the number of cells in each domain is basically evenly distributed among these clusters. So this is a very good feature of our method. And similarly, when we specify the number of cell types, even though the total number of detected clusters is large, but we can find that the proportion of cells in those redundant clusters are very small compared to the other methods. So in general, it's doing a very good job. So given these results, we have a practical guidance on selecting this number of spatial domains and number of cell types. So we usually tell our users to like over-specify the number, so that if you see like for a spatial domain, the number of cells is very small, so it's likely indicate that this domain is redundant, so you don't need to send out large number. But it's kind of a little bit tricky for selecting the number of cell types. So because sometimes if the number of cells of a specific cell type is small, you don't know if that cluster is redundant or they are just real cell types. So caution needs to be made when dealing with cell types. We need to like check for example, check the expression or some marker genes of those clusters that have very small number of cells to make sure that they're either real cell types or just redundant clusters. And these are basically the simulation results, and I will illustrate one example using real data, like how our method performs.
There's a question online from Abdullah who asked, how does your method handle non-single cell resolution data?
Yeah, it's a bit tricky to analyze non-single cell data. We have one, it's in my later slide. We did apply our method to non-single cell resolution data. It's a text DCMB data. I think because it's non-singular resolutions and each spot is composed of multiple cells, so we no longer have the cell type resolution. We no longer have the cell type interpretation. So when we analyze these type of data, it's better to just focus on the spatial domain analysis. Yeah. So the real data we analyzed is a mouse hypothalamus data from the Murphy's technology. And this region is responsible for controlling social behaviors, such as reproduction and circadian rhythms, and homeostatic functions, such as the neural endocrine and cardiovascular regulation. And we obtained a total of 5 tissue sections, and each tissue section has around 6,000 single cells and around 155 marker genes, because this is based on fish technology, so it can only measure a small set of genes. For each of the tissue section, it has 8 symmetric spatial domains, and as illustrated here by this HE image, we can see the structures of these domains, and this is from the mouse Allen brain atlas. So for spatial domain detection, we can find that our method did a very good job at detecting all the 8 symmetric spatial domains. And if we compare our method to the other methods, our method and HMRF, they both detected very relatively smooth spatial domains. And that's because for HMRF, we just specify a list of many different spatial parameters and pick the one that we feel is most reasonable. But this result indicates that our method can estimate the spatial parameter very well. And in contrast, the Bayes space and SPA-GCN, they can barely detect any smooth domains. And this is because Bayes space, it cannot handle single cell resolution data. It's mainly designed for 10x genomics, those type of data. 10x genome using data. And for SPA-GCN, because it's precipitated by the spatial parameter and because it does not give us the very good value of the spatial parameter, so it does not give us the segmentation of the spatial domains. And if we incorporate all the tissue section together, it further improves the performance of the cell type of the spatial domain detection. As you can see here, we from the Bregman negative 0.04 all the way to Bregman negative 0.24, these tissue sections are collected from the anterior all the way to the posterior mouse hypothalamus. So they are expected to have similar spatial domains and cell type compositions. And indeed, we find that the spatial domains detected by our method are very consistent across these 5 tissue sections. And another important thing we find here is that we can actually detect the structural change across these tissue sections. For example, this PBT region on the top, which is colored by red, the size of the region keeps increasing from the anterior all the way to the posterior region of the mouse hypothalamus. And this results cannot be found if we analyze one single tissue sample at a time. For example, here, for the Bregman negative 0.09, we cannot detect the PBT region, which is supposed to be colored by red on the top. And for this tissue section, Bregman negative 0.19, we cannot detect the PV region, which is supposed to be colored by light blue and surround the third ventricle here. So these results highlight the importance of doing a multi-sample analysis and integrate all these tissue sections together to perform the analysis. Just like, so another benefit of an accurate spatial domain detection is that we can use those labels to do to find marker genes, to find domain marker genes, just like what we are doing with cell types. To find cell type marker genes, we can use the spatial domain labels from our method and perform a differential expression analysis to find those marker genes. And by associating these marker genes with these spatial domains, we can better understand the biology underlying these spatial domains. Here I'm giving 2 examples of the genes of the domain marker genes we found. So this SLC18A2 gene is a domain marker gene we found that are differentially expressed in the periventricular hypothalamic nucleus. And this gene encodes a transmembrane protein that regulates dopamine. And we know that the inhibitory effects of dopamine, dopamine has inhibitory effects on the gonadotrophin secretion. And this gonadotrophin is a type of hormone that triggers the expression of 2 other hormones. And those hormones, they further act on the testes and ovaries and can have functions on the reproduction. And so, according to some previous research, this region is well known for regulating the release of this gonadotropin releasing hormone. So our analysis actually indicate this gene SLC18A2 might underline the function of the PB. And another good example is that is this CALCR gene. So this is a domain marker gene for the medial preoptic nucleus. And the CALCR encodes a high affinity receptor for the peptide hormone called calcitonin. And the amylin calcitonin receptor signaling has been reported to mediate the affiliative social contacts. So this is the information we have about this gene. And at the same time, according to some previous study, we know that this region has a regulatory function in reproductive and parental behavior. So now we can like associate. So this connection is suggesting that this function, this gene might underline the function of this region. So besides the spatial domain detection, our methods can also do a good job in cell type clustering. So this is just a UMAP plot from our method from CRET, and we can find that for some cell types, we can probably, for some neuronal subcell types, we can probably identify them. But CRET tend to like aggregate these cell types into the same cluster. And we checked the marker gene of these subtypes. They do have very distinct marker genes. So different cell types have different spatial distribution patterns. So this is a slice. So this is one slice from the mergefish data, and this the x-axis is the x coordinates and the y-axis is the y coordinates. And in each plot, we are showing the one specific cell type, and we can find that for some cell type, they're specifically enriched in certain region, like this receptor neuron 5 is enriched in this PBT region. But for some other cell types, especially those non-neuronal cells, they tend to located across the entire tissue, such as the oligodendrocytes, astrocytes. And this plot, the plot on the right is basically quantifying this observation. For certain regions, like the third ventricle, it's mainly dominated by one cell type, which is the ependymal cells. But for most of the spatial domains, it's a mixture of multiple different cell types. Just like our assumption, we assume a spatial domain is composed of many different cell types. And the enrichment of those cell types might underline the specific functions of those domains. For example, this BST is enriched with the inhibitory neurons, and is consistent with the previous study that this BST region projects inhibitory fibers to the lateral hypothalamus and are important for feeding regulation. And similarly, this FX region, or columns of the fornix, it's a part of the limbic system and enriched with mature oligodendrocytes. And this is also consistent with previous research that continuous oligodendrogenesis in the FX is important for myelination and ensures rapid conduction of neural signals. So this slide is showing more of a technical aspect. So this is showing the posterior estimate of the spatial parameter. So this is in 3 different tissue types, and we can find the estimates are very different. So it does highlight that the importance of estimating this parameter in different tissues in different data sets instead of just fixing it to a specific value. This is the computational performance. So our method is colored in red, and because it's a Bayesian framework, so the computational speed is not like super fast, but the scalability is okay. It scales linearly with respect to the sample size. So for a data set with around 30,000 cells, it takes about 50 minutes. And these are basically all my real data application results. And lastly, I want to briefly introduce our package. So this is the website of our package. So our software is in R package. So the interface is in R, but the underlying functions are written in C++ too. in order to maximize its computation speed. So you can basically reproduce all the results, all the simulation results, and all our findings in the real data in here. So it has an introduction page, software tutorial, and all our simulation real data analysis results. So in the tutorial, it has 4 steps in order to run our package. So our software takes 2 input data, which is a count matrix. It's a list of count matrices because we have many different tissue sections and a list of spatial location matrices. And we also need to specify the number of cell types and spatial domains. And then you create a base object with this input. After that, we only require minimum pre-processing for the data. So the basic pre-process steps include the library size normalization, followed by log2 transformation after adding a serial count of 1. This is very standard pre-processing in single cell data. After that, we select feature genes and perform dimension reduction with PCA. And the default number of PCs we select is 20. Because we are dealing with multiple tissue sections, so we actually also perform the batch effect adjustment with this existing package called Harmony. So this function called base.preprocess basically automates this pre-processing step. And if you want additional pre-processing or you don't like this pre-processing, you can just replace your processed matrix with the running matrix in our object. And after that, you basically run the algorithm. There's not too many options here. And finally, you have to process all the posterior samples from the keep sampling and Metropolis-Hastings algorithm. And there are 2 problems we are going to address in this post-processing step. One is called label switching, because for example, if you are labeling cell type 1, cell type A to be 1, and cell type B to be 2, it's essentially the same to label them as 2 and 1. Label cell type A to be 2 and label cell type B to be 1. So they essentially have the same posterior probability in the distribution. So when you sample them, they can form either of those called posterior mode. So it causes an issue when we try to summarize all these posterior samples because the labeling are not matched. So and this issue is called label switching issue. So in this post-processing step, we use the package called label switching, trying to address this issue. And after that, we just take the posterior mode of all our posterior samples as our estimates. And that's basically the package. And in summary, we have developed a method called BASE for multi-scale and multi-sample analysis of spatial transcriptomics and can perform cell type clustering at the single cell level and spatial domain detection at the tissue region scale with improved performance for both tasks. And the paper has already been published by Genome Biology last year. Please feel free to check the details of the method, and please also feel free to contact me if you have any questions. The software is available at our lab website. And with that, I would like to thank my advisor, Zheng, for his guidance on this project, and thank the audience for listening to my presentation. Thank you.
Another question online from Zheng, who asked if BAS is applicable to DCMB data.
It's applicable. One of our collaborators is actually applying BAS to DCMB data, and the results are quite good. Any other questions?
Any other questions in the room? Give people online a moment in case anyone else is typing. Well, I don't see any other questions, so we'll do another thank you. Thanks everyone for coming, and look forward to seeing everyone next week. Yeah.